Welcome viewers to the UGC Model Syllabus lecture series in Zoology for the third year undergraduate students. Today's topic is Introduction of cloned genes into the host cell. After the gene is cloned into a vector, the recombinant vector has to be introduced into an appropriate host by a suitable method. It should be efficiently and stably be introduced into the host cells or its genome. Genes are the basic hereditary of life. It is our genes that provide the blueprints necessary to produce all proteins in our bodies that ultimately perform the biological functions. Thus, when a gene is stably introduced into a target cell, the gene is expressed and the desired protein is produced. Gene transfer technologies were originally developed as a research tool for investigating gene expression and function. However, as new gene transfer technologies were developed and old technologies refined, the potential applications have expanded dramatically. Currently, there are a number of gene transfer technologies available which vary greatly in the efficiency of gene transfer and the types of cells they are capable of delivering the recombinant vector or genes. The various methods for this purpose include transformation, electroporation, transduction, particle gun, liposome mediated gene transfer, etc. The first method is transformation. The phenomenon of transformation was discovered by Fred Griffith in 1928. Transformation is the uptake of a naked DNA molecule or fragment of DNA by a cell from the medium and the incorporation of this molecule into the recipient chromosome in a heritable form. In nature, when bacteria lyse, they release considerable amounts of DNA into the surrounding environment. These fragments may be relatively large and contain several genes. If a fragment comes in contact with a competent cell, a cell that is able to take up DNA gets transformed. The DNA can be bound to the cell and taken inside. The transformation frequency of very competent cells is around 10 raised to minus 3 for most genera when an excess of DNA is used. This means only one cell in every 1000 will take up and integrate the gene. Competency is a complex phenomenon and is dependent on several conditions. Bacteria need to be in a certain stage of growth. For example, Streptococcus pneumoniae becomes competent during the exponential phase when the population reaches about 10 raised to 7 to 10 raised to 8 cells per ml. When a population becomes competent, bacteria such as Streptococcus pneumoniae secrete a small protein called the competence factor that stimulates the production of 8 to 10 new proteins required for transformation. Natural transformation has been discovered so far only in certain genera including Streptococcus, Bacillus, Thermoactinomycetes, Haemophilus, Neisseria, Moraxella, Acinetobacter, Azotobacter, Helicobacter, Pseudomonas, etc. Gene transfer by this process occurs in soil, aquatic ecosystems and may be an important route of genetic exchange in biofilm and other microbial communities. Many species like E. coli are not naturally competent for transformation. Fortunately, it can be made competent by certain chemical treatment. Two common techniques include electrical or heat shock and treatment with calcium chloride. Both approaches render the cell membrane more permeable to DNA. Both have been used to make artificially competent E. coli cells. To increase the transformation frequency with E. coli, strains that lack one or more nucleases are used. These strains are especially important when transforming the cells with linear DNA, which is vulnerable to attack by nucleases. It is easier to transform bacteria with plasmid DNA, since plasmids are not as easily degraded as linear DNA fragments and can replicate within the host. Transgenic plants are obtained by stable integration and expression of foreign gene into the plant host. Plant transformation can be done for two purposes. One, for giving a particular trait to improve its agricultural, horticultural or ornamental value, and second, for learning about a particular biological process. The traits of interest are usually governed by a single gene or a small cluster of genes. For example, herbicide tolerance, insecticidal resistance, improved nutritional quality, altered flower pigmentation, etc. This is achieved by using plant cell protoplast for transformation. The second mode of bacterial gene transfer is transduction. It is a frequent mode of horizontal gene transfer in nature and is mediated by viruses. Transduction is the transfer of desired genes by viruses. In nature, bacterial genes are incorporated into a phage capsid because of errors made during the virus life cycle. The virus containing these genes then injects them into another bacterium, completing the transfer. There are two different kinds of transduction: generalized and specialized. Joshua Lindbergh and Norton Zinder discovered generalized transduction in 1951. It occurs during the lytic cycle of virulent and some temperate phages and can transfer any part of the bacterial genome. As shown in the figure, during the assembly stage, when the viral chromosomes are packaged into protein capsids, random fragments of the partially degraded bacterial chromosome also may be packaged by mistake. Because the capsid can contain only a limited quantity or length of DNA, the viral DNA is left behind. The quantity of bacterial DNA carried depends primarily on the size of the capsid. The P22 phage of Salmonella enteric Serovar typhimurium usually carries about 1% of the bacterial genome, while the P1 phage of E. coli and a variety of gram-negative bacteria carries about 2 to 2.5% of the genome. The resulting virus particle often injects the DNA into another bacterial cell but cannot initiate a lytic cycle. This phage is known as a generalized transducing particle or phage and is simply a carrier of genetic information from the original bacterium to another cell. As in transformation, once the DNA has been injected, it must be incorporated into the recipient cell's chromosome to preserve the transferred genes. The DNA remains double-stranded during transfer and both strands are integrated into the genome. About 70 to 90% of the transferred DNA is not integrated, but often is able to survive temporarily and is expressed. Abortive transducts are bacteria that contain this non-integrated transduced DNA and are called partial diploids. In specialized transduction, the transducing particle carries only specific portions of the bacterial genome. It is made possible by an error in the lysogenic life cycle of phages that insert their genomes into a specific site in the host chromosome. When a prophage is induced to leave the host chromosome, accession is sometimes carried out improperly. The resulting phage genome contains portions of the bacterial chromosome, about 5 to 10% of the bacterial DNA, next to the integration site, much like the situation with F prime plasmid, which is shown in figure. A transducing phage genome usually is defective and lacks some part of its attachment site. The transducing particle will inject bacterial genes into another bacterium, that is the recipient. Though the defective phage cannot reproduce without assistance of helper phages, the bacterial genes may become stably incorporated under appropriate circumstances. The best studied example of specialized transduction is carried out by the E. coli lambda phage. The lambda phage inserts into the host chromosome at a specific location known as attachment or at sites, as shown in the figure. The phage at sites and bacterial at sites are similar and can complex with each other. At site for lambda is next to the gal and bio genes on the E. coli chromosome. Consequently, specialized transducing lambda phages most often carry these bacterial genes. The lysate or product of cell lysis resulting from the induction of lysogenized E. coli contains normal phage and a few defective transducing particles. These particles are called either lambda D gal because they carry the galactose utilization genes, or lambda D bio if they carry the bio gene from the other side of the at site. Because these lysates contain only a few transducing particles, they often are called low frequency transduction lysates or LFT lysates. Whereas the normal phage has a complete at site, defective transducing particles have a non-functional hybrid integration site that is part of bacterial and part of phage in origin. Integration of the defective phage chromosome does not readily take place. Transducing phages also may have lost some genes essential for reproduction. Stable transductants can arise only if there is a double crossover event on each side of the gal site. The normal phage integrates, yielding two bacterial or phage hybrid at sites, whereas the defective lambda D gal phage can insert as shown in the figure. The transductions are unstable because the prophages can be induced to excise by agents such as UV radiation. Excision, however, produces a lysate containing a fairly equal mixture of defective lambda D gal phage and normal helper phages. Because it is very effective in transduction, the lysate is called a high frequency transduction lysate or HFT lysate. Reinfection of bacteria with this mixture will result in the generation of considerably more transductants. LFT lysates and those produced by generalized transduction have one transducing particle in 10⁵ or 10⁶ phages. SFT lysates contain transducing particles which have frequency of about 0.1 to 0.5. Electroporation is a process for introduction of DNA into cells by exposing them for a very brief period to high voltage pulses, which is thought to induce transient pores in the plasma lamella. Plant protoplasts or cells are suspended in a suitable ionic solution containing linearized recombinant DNA. The mixture is filled in a cuvette which is then exposed to the chosen voltage pulses and desired number of cycles. Protoplasts are then cultured to obtain transformed cells or colonies and regenerate plants. Electroporation can be used to introduce nucleic acids, proteins and other molecules into suspension of animal cells, plant protoplasts, gram-negative and gram-positive bacteria. The transformation efficiency can be as high as 10⁻10 transformants per microgram of DNA. The electroporation apparatus consists of pulse power supply, cuvettes individually wrapped, sterile and fitted with a cap, and electrodes. Low voltage low pulses produces high rate of transient transformation, whereas high voltage short pulses gives high rate of stable transformation. Transformation frequencies can be increased several folds by following methods: a heat shock to protoplast just prior to electroporation, or the presence of low concentration about 8% of polyethylene glycol, that is PEG, during electroporation can be used. However, some plant species are sensitive to PEG. Electroporation has been used to produce stably transformed cell lines and or plants in several plant species, example tobacco, petunia, maize, rice, wheat, etc. In tobacco, the frequency of transformation is as high as 2 to 8% in presence of PEG. Gene gun or biolistic particle delivery system or biolistics is a method of gene transfer that was invented by John C. Sandord and colleagues in 1984 at Cornell University. It is based on the acceleration of microprojectiles to speeds sufficient for penetration of plant cell walls and membranes. The methodology was first used to deliver DNA and RNA into larger epidermal onion cells with the expression of a chimeric cat gene and the transcription of complete tobacco mosaic virus RNA genome. This system has been used with tissues of gymnosperms and angiosperm plants, animals, green algae and yeast. There are two components in this method, propelling mechanism and second, the type of particle to be accelerated. The microprojectiles used are gold or tungsten particles. DNA or RNA is coated on the surface by precipitation with calcium chloride, spermidine or PEG. Many types of such devices have been experimented, of which Biolistic is the most widely used apparatus. As shown in the figure, it uses gunpowder or helium gas to generate the propelling force. Other types of propelling forces used are nitrogen gas, compressed gas and electric discharge. The whole apparatus is usually kept in vacuum to minimize the velocity loss due to air drag. The major factor which affects the transformation of a specific tissue are the size of microprojectiles, the propelling force and the vacuum created. This device is able to transform almost any type of cell and organelles such as plastids. This method is simple and has been adapted for studying transient expression of foreign genes as well as inducing stable transformation with useful genes in different crop plants like onion, rice, cotton, soybean, sorghum, corn, papaya, etc. Agrobacterium based gene transfer. One of the best examples of introduction of clone gene into a host cell by agrobacterium based gene transfer is Bt cotton, which is obtained to make cotton crop insect resistance. The Bt crop can be obtained by transforming plant cells with the Bt gene obtained from a bacterium called Bacillus thuringiensis. The Bt gene is cloned into a vector called the Ti plasmid vector, which is obtained from Agrobacterium tumefaciens. The Bt gene is cloned into this vector and the recombinant vector is reintroduced into Agrobacterium strains. The vector is then introduced into the plant cell by infection by the Agrobacterium strain. The plant cells that are expressing Bt gene are then selected from the clones that are obtained on the plant tissue culture media. The selected clones in which Bt protein is produced is grown or hardened to get whole plants that expresses the Bt toxin in the leaves and impart resistance to insect pests. Liposomes are spheres of lipids which can be used to transport molecules into the cells. These are artificial vesicles that can act as delivery agents for exogenous materials including transgenes. They are considered as sphere of lipid bilayers surrounding the molecule to be transported and promote transport after fusion with the cell membrane. Cationic lipids are those having a positive charge and are used for the transfer of nucleic acids. These liposomes are able to interact with the negatively charged cell membrane more readily than uncharged liposomes, with the fusion between cationic liposome and the cell surface resulting in the delivery of DNA directly across the plasma membrane. Cationic liposomes can be produced from a number of cationic lipids, example DOTAP, DOTMA, etc. These are commercially available lipids that are sold as an in vitro transfecting agent as lipofectin. Liposomes for use in gene transfer vehicles are prepared by adding an appropriate mix of bilayer constituents to an aqueous solution of DNA molecules. In this aqueous environment, phospholipid hydrophilic heads associate with water, while hydrophobic tails self-associate to exclude water from within the lipid bilayer. This self-organizing process creates discrete spheres of continuous lipid bilayer membrane enveloping a small quantity of DNA solution. The liposomes are then ready to be added to the target cells. Thus, a variety of methods for gene transfer into host cells are available. The method of choice depends on the type of host cell, the efficiency of transformation required, as well as the cost of the process. Thank you very much.